You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is September 15th, 2025. We will be continuing our basic immunology course doing chapters 2 through 4 of Dr. Sompayrac's How the Immune System Works. This is presented by Dr. Nikita Raje. She is an associate professor of internal medicine and pediatrics and is the chief in the section of allergy and immunology at Children's Mercy Hospital in Kansas City.
This is just the introduction slide for the year, the two textbooks that we are going to cover for the year. My disclosures, nothing relevant to the talk today. And then there's some pre-test questions. We'll go over the answers at the end. What DNA segments are the first to rearrange in adaptive immune cells? Which antibody is the best at fixing complement? And what molecule causes the release of the clip or invariant chain in MHC class 2 presentation? I believe that's it. Okay, but we'll start with chapter 2. So we're going to go through chapters 2, 3, and 4. So chapter 2 from Sompayrac textbook is Innate Immune System. And again, I forgot to mention, I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City, and today is September 15, 2025. My email is mentioned here if you have questions after the talk. All right, so this is the outline just for the chapter 2. We'll go through that, and then we'll move on to chapters 3 and 4. So innate immune systems made up of complement, phagocytes, pattern recognition receptors, and dendritic cells, natural killer cells, and then we'll talk about cytokines. So let's talk about the complement system. And please feel free to stop me at any point if you think that you have a question, because everything builds on to the talk, in the talk. So I don't want you to wait till the end if you need to talk to me. All right, so the complement system is made up of various proteins. These are small proteins that come together to form bigger complexes, and they help in killing the pathogens, and they also signal the other immune system players that the attack is on. Where are they formed? They are produced in the during the first trimester of fetal development from liver, and they're ready to go when the child is born. Complement system, like any other part of the immune system, does need activation, and that's just a safety net because any part of the immune system by its immune response can cause inflammation, and the immune response itself is, if directed towards the wrong molecule, can cause problems for the human body. So the complement system also needs activation before it can function. Let's see. All right, there. So there are 3 pathways of complement activation: classical, alternative, and lectin pathway. The classical pathway was so called because it was discovered first. It is dependent on the antibodies for activation. Alternative pathway is actually the more innate form of the complement system, where there is no antibody involvement and it is spontaneous. But was found later on and is called alternative complement pathway. And then the lectin pathway is the smart complement pathway, which means it only binds to the pathogen molecules. That's your lectin, mannose binding lectin pathway. Because we are talking about innate immune system here, we are just going to focus on the alternative pathway here. We are not going to worry about the classical pathway, just because that's part of the functioning of it involves antibodies, which is the adaptive immune system. So alternative pathway, how it works is there is a complement fragment called C3 that is continuously and spontaneously cleaved to smaller parts, C3b and C3a. And so the C3b binds to, as it falls, right, it's continuously cleaving, and the C3b particle will bind to the amino or hydroxyl groups. So the amino and hydroxyl groups would be found in proteins or carbohydrates, which are present in the bacterial cells. So these C3b are like grenades, right? They are just falling. See, they are falling continuously, and if it comes across amino or hydroxyl group, it's going to bind to that. But it gets 60 whole microseconds to do this job. So in those 60 microseconds, if it is not able to bind to an amino or hydroxyl group, it's neutralized to water molecule, right? It's neutralized by the water molecule. So here's a picture showing, say, a surface of bacterium. So a bacteria is present, the surface of the bacterium will have the amino or hydroxyl group. Complement C3 is cleaving to C3b and C3a, and the C3b can go and bind to that group on the bacterial cell wall. Now that C3b binds to another protein called protein B. And protein B is like scissors. It comes and clips C3b, right? So now C3b bound to protein B can be cleaved so that the protein B is also cleaved to another particle called BB. So the C3b BB will again act as a another form of scissors that cleave C3. But this is a very high functioning scissors that keep cleaving C3 to C3a and C3b very efficiently compared to the spontaneous process. So if the spontaneous process is going on at certain pace, this C3b BB, once it's formed, is gonna cleave the C3 to C3b very quickly and efficiently. And so it's called C3 convertase. As more and more molecules of C3 are cleaved to C3b, another C3b molecule comes and binds to that C3 convertase to form the C5 convertase. So now this complex C3b BB C3b will now be able to cleave the molecule C5 to C5a and C5b. And so that's when the C5 complement, which is the later part of the complement system, gets involved. So these two molecules are the C3 and C5 convertase, which are a little different from the other pathways, but the process is the same. The complement fragments come together, form a complex that is called C3 convertase that cleaves more C3 molecules, finally ends up making C5 convertase, and then the C5 convertase cleaves the C5 molecule. The C5 molecule can then attract C6, 7, and 8, and all of them together form a stalk in that bacterial cell wall. And once that happens, there are multiple molecules of C9 that come and attach to that stalk to form a hole in that cell wall. That's called membrane attack complex. So that's your terminal complement pathway after the alternative complement pathway activation. All right, but like any other part of the immune system, again, we need some safety net so that if the complement pathway gets activated, so as we know, alternative pathway is always, it's spontaneous and it's continuing to cleave that C3 molecule to C3b. So we need some other safety nets as well to make sure that if it gets activated and is not needed, there are certain, and it's trying to attack human cells, then there are certain processes and safety factors that are available. So those proteins are called membrane cofactor protein MCP, CD46, decay accelerating factor or CD55, and protectin CD59. As you see here, the overall immune system, this is the theme throughout immunology as we go through these chapters, that molecules have two names. It has the name that's how it was found, some kind of functional aspect of that helped in naming the molecule, but also it has a CD number, and those are interchangeable, but the unfortunate part is that you have to memorize both of those. So membrane cofactor protein, MCP or CD46, will clip the C3b to its inactive form. Decay accelerating factor accelerates the destruction once the C3b BB is formed, which is the C3 convertase. and then say everything goes through, the C5 is cleaved, the stalk is formed, protectin or CD59 will prevent incorporation of C9 into membrane attack complex. So what happens if there is a defect in either CD55 or CD59? Does anyone know? Anybody?
Is it PNH disease?
PNH is correct. Paroxysmal nocturnal hemoglobinuria is caused by defect in these molecules. All right. The second one is lectin pathway. So remember, the alternative pathway is like grenades. C3b is continuously being cleaved, and C3b can continue to fall like grenades and pick up any of those amino or hydroxyl groups. Lectin pathway, not so much. These are smart bombs, right? So the lectin is a protein that can bind to a carbohydrate. That's all it means. Lectin means a protein that can bind to a carbohydrate. Mannose binding lectin means the protein can bind to the carbohydrate, which is mannose. And mannose is found on very specific pathogens like Candida, or some viruses like flu A, HIV, some bacteria like Salmonella, Strep, Leishmania. So mannose binding lectin is also, again, like alternative complement proteins, is produced in liver. And it is specific for the mannose, the sugar that is found in specific microbes. How does this pathway function? The mannose binding lectin can bind to another protein called mannose binding lectin associated serine protease. So MASP is an enzyme, right? So it has a capacity, it's a protease, so capacity to cleave a protein. So when mannose binding lectin comes across mannose, it will grab that pathogen because it has mannose in it. Now the MASP that is bound to the MBL can clip the C3 to C3b. Once that happens, the rest of the pathway is the same. So C3b can then bind to protein B. Protein B can make BB and bind to C3b to form that C3 convertase, and so on and so forth, and then makes the C5 convertase. So C3 and C5 convertases then lead to the membrane attack complex that we just talked about. So this is mannose binding lectin pathway. Now thinking about the complement system overall, what's the function? Of course, it makes that membrane attack complex that helps in killing the microbe. Apart from that, it has a couple of other roles. It can help in opsonization. Remember, opsonization means it tags the microbe and it makes it ready for phagocytosis. So opsonization is a function of a fragment of C3b called IC3b, and IC3b can opsonize the pathogens for phagocytosis. There are other fragments when the C3b or C5b are created. The smaller fragments C5a and C3a are also produced, which are chemoattractants, which means they send a signal that we need help and attract other cells to the site of infection. So they attract neutrophils, macrophages, and that's how then that phagocytosis will help for those opsonized microbes. So C5a, C3a are also called anaphylotoxins. All right, so that's our complement pathway, part of the innate immune system. The classical pathway is what we'll talk about with adaptive immune system. Now we move on to another part of the innate immune system, and we'll talk about the phagocytes. So what are the two professional phagocytes? Macrophages and neutrophils. The difference? Macrophages are sentinel cells, which means they are in the tissue, they are below the surface under skin or mucosa, and they are there so that they can watch out for any incoming invaders. Compared to neutrophils that are mostly in the circulation, and they are like foot soldiers. They are called upon, and when called upon, they go to the site of infection, they are killing the microbes, they are doing their job, and in the process getting killed themselves. Let's talk about the macrophages. Macrophages are produced as monocytes in the bone marrow. They enter the tissue and reside there as macrophages, but in the tissue, they are present in three different states. They are either resting, right, or activated or hyperactivated. So let's see the pictures. So macrophages are in resting state most of the time for months, right? And they are pretty much garbage collectors, which means they're just going to move around and see what's around them. They're going to grab some things from the environment that they are in. And really, they are just kind of there, watching out for any invaders. And they express very few class 2 MHC. So remember, macrophages are one type of antigen presenting cells for T cells. But at this resting state, they are not really doing any presentation to T cells, so they really don't need that class 2 MHC on their surface. So pretty much they are just resting and then they're not in a mood to kill. However, when activated, what activates the macrophages? Nothing like interferon gamma. So remember, macrophages, for their activation, the most important signal for that is interferon gamma. So the interferon gamma is typically produced either by the adaptive immune system, so helper T cells, or in the innate phase of the infection, the NK cells also produce a lot of interferon gamma that comes in handy because it is going to help in activation of these macrophages. Now that they are activated, they are going to upregulate the class 2 MHC on their surface so they can start doing some work. They can start helping with presentation of antigens to T cells. And they can also kind of start engulfing the invaders around them. So remember, interferon gamma makes these macrophages annoyed or activated, and they're going to start making class 2, upregulate their MHC class 2 for presentation, and they're also going to engulf things from around. But in their hyperactivated state is where they are absolutely at work, they are busy, they are activated directly by the invader. So interferon gamma plays a role, but an invaders and the signals from bacteria or any other microbes such as LPS or manos, those are the signals that once the macrophage comes across that, it is absolutely sure that there is a problem. And so now they start growing, they stop proliferating, they secrete TNF, they engulf large particles as big as a unicellular parasite. They will have a lot of class 2 MHC on their surface, and they'll also make a lot of enzymes that will come in handy after they phagocytose the microbes for killing those microbes. So lysosomes and reactive oxygen species. So that's how the stage of macrophage changes based on what it is coming across. The signals are important, whether it's interferon gamma that activates them a little bit, or signals directly from the invader. All right, moving on to neutrophils. Neutrophils are produced in the bone marrow, about 20 billion, 70% of the white cells. They're short-lived, about 5 days. They are different. They are phagocytes like macrophages, but unlike macrophages, they are not antigen-presenting cells. They are professional killers. They are on call 24/7. They are in the circulation, and once they are called upon, they'll exit the blood circulation, and within 30 minutes, they are fully activated. They go to the site of infection, engulf invaders, and kill. There are chemicals that can liquefy both the cells and the connective tissue that are released from the neutrophils, so they kind of create that, they produce that chemical that gives rise to that liquefaction, and that produces the pus. They also produce cytokines like TNF. And then apart from the phagocytosis and killing of the invaders inside the phagolysosome, they also form what is known as neutrophil extracellular traps, or nets. Nets, which means that the neutrophils extrude instead of engulfing the microbe and putting it in phagolysosome where the enzymes are going to work and kill that microbe. Sometimes the microbe is very big, and neutrophils are like, well, we can't do anything because we can't engulf or phagocytose the microbe. In those cases, they will extrude their DNA and the enzymes onto that area, so that creates a neutrophil extracellular traps where the microbes can be trapped and killed because of the enzymes. So for these neutrophils, and other white cells also use this system, but we'll talk about the neutrophils for now. So remember, neutrophils are like fellows on call. When you are on call and you say you are tired going home, you are on the freeway and you get a call and you get a page to say you're needed at the hospital. What are you going to do? You are going on the freeway. You're going to slow down. So you're going to move to the peripheral lane, and then kind of apply some brakes and then take the exit. Same thing with the neutrophils. They are on call 24/7. They are in the circulation in the fast lane. So at that fast pace, they are not going to be able to come out of the circulation. But they have to apply some brakes when they are called upon. They have to move to the peripheral lane, apply brakes, and then come out and exit the circulation. So for that, there are several mechanisms so that they can apply those brakes. So neutrophils have something called selectin ligands on their surface. And the selectin ligands can bind to the selectins on the surface of the blood vessel. Besides that, they have integrins, another surface molecule on their surface called integrins. And the integrins can bind to the integrin ligands or ICAMs on the surface of the blood vessel wall. So selectin ligands and integrins are on neutrophils, and integrin ligands and selectins are on the blood endothelial wall. All right, so say there is an infection. That infection site is going to release cytokines like TNF. Remember, the macrophages are doing their job, releasing some TNF, maybe IL-1, and that will help in endothelium expressing selectin. So initially, there is no selectin. On a routine basis, there is no reason for neutrophils to slow down, and they are going fast in the circulation. But when there is a signal of infection, selectins are expressed on the surface of the blood vessel wall, and that comes in contact with the selectin ligands on the neutrophils. So it's almost like Velcro. It kind of binds to it and then gets released. And so it moves a little forward and then kind of starts rolling on the surface of the endothelium. There are other signals like C5a, LPS that cause neutrophils to express integrins. So at baseline, there's no selectin on the endothelial wall and there are no integrins on the neutrophils. So now that the integrin is expressed on the neutrophil surface, it will come in contact with these ICAMs and come to a complete stop. So as it kind of rolls with the selectins, when it makes contact with the integrin ligands, it will come to a complete stop, and then it's gonna exit the circulation. And the reason it follows the scent of some other molecules called FMET or C5a. So these help neutrophils to tell them where to go. So it's almost like a GPS, right? Neutrophils don't know, they come out of the circulation, but where do they go? And FMET and C5a act as GPS to lead them to where they need to go. All right, so, and then on their way, while they are being they are activated by the cytokines like TNF, and as they move to the site of infection is where they're going to give rise to that inflammation. All right, so that's phagocytes. Now we talk about the pattern recognition receptors. Any questions so far? All right, let's talk about pattern recognition receptors. Pattern recognition receptors are meant to recognize danger signals. So what are these danger signals? They are either associated to pathogens or because of some damaged tissue. So they are called pathogen-associated molecular patterns or PAMPs, or damage-associated molecular patterns called DAMPs. So PAMPs and DAMPs is what the PRRs or pattern recognition receptors recognize. So once the pattern recognition receptors recognize these danger signals, they help in activation of the innate immune cells. So there is maybe activation of macrophages. Macrophages release cytokines, and that helps in that inflammation and the innate immune response. The what you have to remember is that the pattern recognition receptors recognize the general characteristics of a class of invaders. And typically these structural patterns that they recognize are indispensable to the pathogen, so that they are not altered by mutation. So over time, the PRRs don't change because they are recognizing the typical pattern that is needed for survival of pathogens for millions of years. So that doesn't change over time. I don't know what's going on. Okay. So among these pattern recognition receptors, the most well studied are toll-like receptors. And there are about 10, that are described more in detail, but there might be more. And these toll-like receptors can be in different places related to the cells. So these are cell-associated pattern recognition receptors, and they are either on the surface, say like TLR4, or inside the cell, which are intracellular or on the membranes of phagolysosomes, such as TLR7 and 9. So depending on where they are and depending on the type, they recognize specific PAMPs or DAMPs. So for example, toll-like receptor 4 is specific for recognizing or attaching to PAMP such as LPS, amongst other PAMPs. And then TLR7, for example, binds to single-stranded RNA. Single-stranded RNA is typically from, say, viruses like HIV or influenza. TLR9 binds to double-stranded DNA from bacteria or HSV. Apart from the toll-like receptors, toll-like receptors, especially the intracellular one, are really important for recognizing viral infections. Another innate defense against viruses includes a system called interferon system. So remember, when we talk about dendritic cells that are sitting and are working like sentinel cells in the tissue, those are different type of dendritic cells. Here we are talking about another type of dendritic cells called plasmacytoid dendritic cells. So plasmacytoid dendritic cells are factories of interferon. Their job is to make interferons. What they do is, when, say, toll-like receptors like 7 and 9 detect viral RNA or DNA, these plasmacytoid dendritic cells will get activated in about 6 hours of that, and then they will produce 1,000 times more interferon than any other cells. The interferon that they produce, typically type 1 interferons, alpha and beta, and that helps in slowing down the viral replication, because, as the name suggests, interferon interferes with the viral replication. How does it work? If there is a viral infection, here is an infected plasma dendritic cell. It produces interferon alpha and beta. If it is an infected, if it basically goes and acts on the cell, warned cell. And then if it is an infected cell, it will make that cell undergo apoptosis. If it is a cell that is uninfected, it continues business as usual. However, it has been warned of the viral infection. So it helps in reducing the replication of viruses. And for the uninfected cells, the goal is to protect it from that infection. All right, we are moving on to talking about NK cells. NK cells are one type of lymphocytes. They mature in bone marrow like other lymphocytes, but their DNA does not rearrange over time, and it does not adapt to the microbes that it comes across. So NK cells are the lymphocytes that are part of the innate immune system. Their half-life is about 1 week in absence of infection. So they are also in circulation or tissues. In the circulation, they'll use the same roll, stop, exit strategy that we talked about for neutrophils. So they are going to use those selectins and selectin ligands, integrins and integrin ligands, so that they initially roll and then come to a complete stop and exit and then move towards the site of infection. One of the big function of natural killer cells is to kill, And the way it kills is that it comes across a cell. When it interacts with the cell, it uses two kinds of receptors. One is to bind to the target cell, whether it can bind to it, right? And the other is, so that's the activating receptor, and then the other is the inhibitory receptor, which is looking for any signal that tells it that it's a normal human cell. How does it know? Class 1 MHC is expressed by all nucleated cells, human cells. And so if it comes across class 1 MHC, then the NK cell gets a signal saying, don't kill. So don't kill this cell because this is likely a normal human cell. If this inhibitory signal does not come across this molecule, then it realizes that it's likely not a normal human cell. Either it's damaged or it is, say, a cancer cell, and in that case, it is going to kill that cell. That's how the natural killer cells kill their target cells. Apart from that, NK cells can release inflammatory cytokines. Remember, we talked about how NK cells can release interferon gamma that helps in activation of macrophages. So they also produce IL-2 that helps in activation of T cells. So they are big producers of these cytokines that help in activating other immune cells. We already talked about these. The target cells are either cancer cells, virally affected cells, or other infected cells. We talked about the activation and inhibitory receptors, and how does it kill? So the killing is done by two different ways. One is called the perforin granzyme system, which means it releases the granules into that target cell called granzyme B, and that those can help that cell undergo apoptosis. The other way of killing the target cell is using Fas ligand on the NK cells. Fas ligand binds to a signal called Fas on target cells. Any target cell that expresses Fas is basically saying, come and kill me, right? And the Fas ligand on the NK cells recognizes that signal and will help that cell undergo apoptosis. So we talked about cytokines, we talked about toll-like receptors, we talked about macrophages and NK cells. This is just showing that the parts of the innate immune system that interact with each other, especially the NK cells producing interferon gamma, macrophages that release TNF, and they are helping each other in their job. This is the overall innate immune system, apart from just the macrophages. Macrophages are in inactive state, but when activated, they will release cytokines, and they will also help in killing the microbes. Natural killer cells, again, will release cytokines that help in activation of macrophages, and the activated NK cells will kill the microbes or kill the target cells. Interferon alpha and beta are types of cytokines that help in fighting that viral infection as well. Here is the complement system that's helping in fighting different kinds of infections. So that's the end of chapter 2. Questions? All right, moving on to chapter 3, we are talking about B cells and antibodies here. Remember, now we are talking about adaptive immune system. B and T cells adapt by rearranging their DNA. We talked about the VDJ recombination last time. And remember that adaptive immunity has this special feature of memory for infections. B cells made in bone marrow, they have this B cell receptor on their surface. It is nothing but a molecule of IgM, but this IgM has specificity for a certain antigen. This B cell receptor, or BCR, is made up of IgM, so it looks like IgM. It has a heavy chain, it has light chain, it undergoes gene rearrangement. It uses this rearrangement where the D segment joins the J segment, and once that's done, then the V region rearranges as well, so DJ comes before VDJ. When there is a productive rearrangement, which means that that rearrangement is successful, it can make a B cell receptor, and each of that rearrangement for different B cells can make different BCRs. So there are diverse BCRs on surface of various B cells. Naive B cells. What are naive B cells? What does naive mean? Naive means that it has not come across the antigen that is specific for that B cell. So naive B cells have not yet encountered that antigen, and the antigen that is specific for that particular B cell is called cognate antigen for that B cell. Now remember, if that naive B cell comes across its cognate antigen, the B cell will have the BCR, and it binds to a small portion of that antigen. Not even the whole microbe. We are talking about one antigen from the whole microbe, and part of that antigen to which the B cell receptor binds is called epitope. So once the BCR binds to the epitope of the antigen, it sends downstream signals. And for this downstream signaling, it has these molecules on it called Ig alpha and Ig beta. When that signal goes through, that's when it can get activated. So the signal requires that either several BCRs on the surface of the cell can bind to multiple similar molecules or epitopes, right? That's called cross-linking. That's typically found in polysaccharides because it's the repeating sugar molecules that are next to each other. The best example of that is pneumococcal bacteria. It has that polysaccharide capsule on its surface, and it can bind and cross-link the B cell receptors on the surface of a B cell, and that's how it gets activated. Or if there is no cross-linking, then it needs a second signal. The second signal is, we'll talk about that in a little bit here. Let's see. So here is the cross-linking shown where the antigen has repeat epitopes and several BCRs come close to each other to cross-link. Here is a picture of the BCR. Here is the B cell. B cell has the IgM molecule with heavy chain and two heavy chains, two light chains on its surface. When the BCR binds to the epitope, it will send downstream signaling through Ig alpha and Ig beta inside the cell. All right, the other type of activation instead of cross-linking is using co-receptor and second signal. So the co-receptor binds to the same antigen a different epitope. So the co-receptor, or not even epitope, sorry, it binds to a complement fragment. So say the antigen is opsonized by a IC3b and the BCR binds to the epitope on the antigen, the CD21 complement receptor will bind to the complement fragment that is used to opsonize this antigen. So the co-receptor is CD21. Remember, CD21 is used by EBV for its entry. So CD21 is the co-receptor of B cells, and it binds the complement fragment. The T cell dependent antigen will require a help from T cells, and the T cells provide that co-stimulatory signal. Anyone know what that is? For B cells? CD40 ligand. So, and then the other one is T cell independent. We saw the cross-linking, which is independent of the T cell. So activation of B cells can occur in two ways: T cell dependent and T cell independent activation. Once it's activated, it will help use that signaling through Ig alpha and Ig beta. The T cell dependent signaling requires that co-stimulatory or second signal provided by CD40 ligand. CD40 ligand is on T cells. T cell independent, we talked about cross-linking, and then there is something called polyclonal activation. So BCR independent. So the B cell receptor has nothing to do with the activation of B cell in that case. And that's done by what we see in vitro when we do mitogen antigen tests. When we send those in, those B cells are activated by mitogens, plant products that will cause BCR independent activation, which means it doesn't even have to be a particular type of BCR. It does not have to be specific to one antigen. All the B cells of different specificity can get activated at the same time. All right. Once that signaling goes through after activation, the B cells can undergo various processes or maturation processes. They can class switch or they can undergo somatic hypermutation, and there is a career decision to make. So let's look at that. Class switching. So the first antibody that's made by B cells when they are activated is IgM, and um, when this IgM is supposed to the B cell is undergo class switching, which means from IgM it needs to switch to GA or E. At that time, remember the VDJ part is bound to the constant part called C. CM or C mu is the one that makes the IgM, and that C mu is followed by DGEA. So depending on what type of infection it is it a parasitic infection? You need IgE. It's going to cut out that portion between CM and CE and cut out that portion so that the CE gets attached. If it's a bacterial virus infection or a viral infection, you need IgG. It's going to cut out CM and CD, and the CG gets bound to the VDJ. If it is, say, a mucosal infection where you need IgA, it will cut out the rest of the portion and the CA gets attached to the VDJ. That's how the class switching works. Talking about IgM itself, different classes. So depending on the constant region, there are different classes of immunoglobulins that these B cells can make, and they have their own specific properties. IgM is a pentamer, which means 5 molecules of IgM. Can you see my pointer? Can come together and are joined with a joining chain here to form that pentamer. And it fixes complement very well. So that's the best one for complement fixation. IgG, there are various subclasses 1 to 4. It can also fix complement 3, 1, 2. Those are so IgG3 better than IgG1, better than IgG2 for fixing complement. The other functions of IgG are opsonization. It helps in antibody-dependent cellular cytotoxicity. It can cross placenta. Its half-life is about 3 to 4 weeks. And then the class switching to IgG occurs in presence of interferon gamma. IgA, it's the most abundant antibody in the body. IgG is the most abundant antibody in the blood circulation. IgA guards the mucosal surfaces, and it usually is in the form of a dimer, which means 2 molecules of IgA are attached by that joining chain or clip. And that clip helps, it facilitates the transport of IgA across the mucosal wall. And it also helps in making the IgA molecules resistant to the GI enzymes. IgA can be secreted in breast milk, and then class switching to IgA occurs in response to presence of TGF-beta cytokine. IgE causes mast cell degranulation. It helps in defense against parasites. It causes allergies, gives us business. Class switching is induced by IL-4 and IL-5. B cells can also undergo somatic hypermutation. So remember, class switching occurs in that constant region, C region of the immunoglobulin. Somatic hypermutation occurs in that FAB region of the immunoglobulins. So the BCRs mutate so that they rearrange the antibody gene that codes for FAB region in order to change the affinity for invader. So say it binds to the flu virus, and the flu virus this year, now the antigens have changed for this year's season. Now the B cells are going to go back and see if they can make better antibodies for this year when it comes across that virus, so that they can bind with higher affinity for that virus. So as they undergo somatic hypermutation, the affinity improves. And then the last one is career decision, where the B cells decide if they want to be plasma cells or they want to be memory B cells. So plasma cells are the ones that will produce a lot of antibodies right then and there. Memory B cells are going to be staying for a longer time, and when needed, they can produce B cells or plasma cells whenever they are needed. So those are the processes for the B cells. And that's about it for B cells. Any questions? Okay, All right, moving on to chapter 4, the magic of antigen presentation. So, let's talk about these antigen presentation molecules, class 1 and class 2 MHC. Remember, they act like billboards on the surface of the cell, showing off whatever is going on in the cell. Whatever antigens or proteins are produced in the cell, not produced, sorry, are present in the cell, whether they are produced in the cell or brought from outside to the cell, that antigen is going to be displayed on the surface using class 1, class 2 MHC billboards so that they can present that antigen to the T cells. That's your antigen presentation. Class 1 MHC, it has a closed groove. Remember, we talked about this. It has a closed groove because it has that structure of beta 2 microglobulin chain binding to one heavy chain. And so that forms that closed groove where 11 amino acid long peptide can stay. Class 1 MHC is different types, HLA type A, type B, type C, HLA A, B, C. And then it has that chain, beta 2 microglobulin. Class 1 MHC is found on all nucleated human cells. How does the antigen presentation work in the class 1 MHC pathway? So remember, class 1 MHC is present on all nucleated cells, liver cell, muscle cell, bone cell, all the cells have class 1 MHC on them. They present endogenous proteins, which means if the protein is synthesized inside the cell, these proteins are cut in the system called proteasome. Here's that green thing, that's a proteasome. It's like a machine to cut that protein into various peptides. The proteasome is very smart, and it can cut peptides in a way that the amino acids are at the end are very specific. So these peptides are then carried from the proteasome inside the endoplasmic reticulum. They are carried by a vehicle that is known as TAP. Once the TAP transports the peptide to the endoplasmic reticulum, in the endoplasmic reticulum are freshly made class 1 MHC molecules. Those come and the TAP is bringing in the peptides, and the class 1 MHC gets loaded on with that peptide. Once these MHC class 1 are loaded with the peptide, they go through the Golgi apparatus and then come to the surface of the cell, where they are going to express that class 1 MHC bound to the peptide on the surface to present it to the T cells. That's your class 1 MHC pathway. Remember, proteasome, TAP, endoplasmic reticulum, where the MHC class 1 gets loaded, then goes through Golgi apparatus and to the surface of the cell. Class 2 MHC, little different. It has an open binding groove. It can take up to 13 to 25 amino acid long peptide, and it is present only on antigen presenting cells. So only the professional antigen presenting cells have class 2 MHC on them. These are HLA-DP, DQ, DR, right? So these are important where they are gonna display exogenous proteins, which means these proteins are not synthesized inside the cell. These are the cells that have class 2 MHC are macrophages, B cells, and dendritic cells, right? These cells are pretty much bringing in proteins that are produced outside inside the cell. So they express these cells can use class 2 MHC on their surface to display those exogenous proteins. Class 2 MHC is formed by alpha chains and beta chains. They initially bind to something called invariant chain. Because I'll show you in a picture. Sorry, let's just move on to prevent the protein binding until it's in the cytoplasm. And then something called, well, we'll come back to that. So here is our endoplasmic reticulum. Again, the proteins are produced in endoplasmic reticulum. So class 2 MHC is also produced in the endoplasmic reticulum along with something called invariant chain. So the invariant chain closes the groove for MHC class 2 complex when it is produced. Why is that? Otherwise, in the endoplasmic reticulum, the tap that brings in peptides for class 1 MHC will get loaded onto class 2 MHC. That's not what we want. So the class 2 MHC is covered by invariant chain. It's like a lid. The lid is called invariant chain, so it is closed off. Now the empty class 2 MHC goes through the Golgi apparatus and goes into something called an endosome. Now remember, the dendritic cells, macrophages will bring in, or so like particularly macrophages will have that phagosome. It's going to engulf proteins from outside, bring it in, and those are going to be cut up into peptides. The endosome and the phagosome can fuse with each other, and in that process, when that endosome is going to do that, the invariant chain is going to cut, and only a small portion of that, because it's no longer, the rest of it is no longer needed for transportation because it's not getting transported anywhere. So the part that stays behind and covers, lids off that class 2 MHC now is called clip. And that binds to the clip with the MHC class 2 is now inside that pouch, phagoendosome, is going to be available for the peptides because something comes in and takes the lid off, takes the clip off, so that the peptide now gets loaded onto that class 2 MHC, and then that goes to the surface, so that peptide is displayed on that billboard of class 2 MHC for the T cells to be to look at it, right? So what takes off the lid or the clip is called HLA-DM. All right, so antigen presentation, we talked about the APCs, macrophages, dendritic cells, and activated B cells are the APCs. They require that presentation. Now, what happens with that presentation? So now we know that class 1 or class 2 MHC, especially the APCs, will have class 2 MHC. They will express the peptide on the surface. And remember, class 2 MHC is specific for CD4 cells, helper T cells. So helper T cells come along, TCR can now look at that billboard, class 2 MHC, with the antigen, and that is their first signal for activation of T cells. So the APC did its job as the antigen presenter. Apart from that first signal, helper T cells are part of the immune system. They need a safety net, so they need that second signal. Remember, we talked about safety deposit box and the second key from the banker along with the main key. So here is the main first key that helps in activation, but for the complete activation to occur, that second signal is also provided by APCs. This tells the T cells that there is danger. And so it really needs to get activated. So that is provided by something, one of the co-stimulator is called B7 and binds to CD28 on the surface helper T cells. So we talked about macrophages. Now we talk about dendritic cells. Dendritic cells are also sentinel cells, which means they are in the tissue, ready to be activated if there is an invader coming in. These dendritic cells will get activated by cytokines like TNF or toll-like receptors. And once activated, they take the information of what type of infection. Is it bacterial or viral? Where is the infection? Is it in the toe or is it in the brain? Is it in the liver? It takes that information and goes into the lymphatic circulation, goes to the lymph node. In the lymph node, they meet the T cells. So that's where they present the antigens to the T cells. The dendritic cells are short-lived. That's what they do. They go from the site of infection through the lymphatics to the lymph node, present the antigen, and that's their job. In that process of moving from when they are in the tissue, they are just sampler. They are taking the samples from the outside. When they know, that's before the attack. When there is an attack, so there is a microbe, in that case, there will be cytokines around. So there will be TNF, there will be toll-like receptors that are binding to the pattern PAMPs and DAMPs. In that case, the dendritic cells will start making a lot of class 2 MHC in them, and they start traveling. And they start expressing more B7 for presentation and co-stimulation. So presentation by class 2 MHC and co-stimulation by B7. So they start expressing a lot of those on their surface by the time they reach the lymph node, and so that they can present the antigen and activate and help with co-stimulation for the T cell activation. That's the dendritic cells. We talked about macrophages. They're activated by interferon gamma and TLRs, and they help in re-stimulation of T cells. Activated B cells, they bind to the antigen when there is binding of antigen to the BCR. They engulf the BCR and the antigen together. All of it goes into the B cell. And then they package the antigen onto the class 2 MHC and then present it to T cells. They are really efficient presenters. Remember, it's the activated B cells that are APCs, not regular B cells. Well, apart from the typical class 2 and class 1 MHC, there is something called non-classical MHC. Non-classical MHC, the best example is CD1, and it looks like class 1 MHC, which means it has one chain bound to or paired with beta-2 microglobulin chain. And instead of binding to peptides, remember, T cells, what they look for is peptides, proteins. Protein antigens is what class 1 and class 2 MHC present to T cells. But there is something called CD1 that can bind to lipids, and they can present that to T or NK T cells. And so that's more of the non-classical MHC. All right, that's the last slide. We'll go through a few questions that we saw at the initial time of the presentation. So question 1, what DNA segments are the first to rearrange in adaptive immune cells? Anyone?
The VDJ-CM.
So what comes first?
So DJ comes first.
Oh, that's
DJ.
Yeah.
So DJ before VDJ. So disco jockey before video jockey. So DJ before VDJ. Which antibody is the best at fixing complement?
IgM.
IgM.
And then
IgG.
which
Yeah.
one?
Yeah, we go IgG.
Y. I don't know what's that class of IgG.
Yeah, subclasses.
IgG1 and 2. 100% sure.
Yeah, I think I go IgG1. I'm not positive though.
Okay, so IgM and then 312. That's the area code of Chicago. 312, remember. 312. M and then 312. So if you go directly to 312, that doesn't work either. IgM is the very best, and among IgG, it's 312. What molecule causes the release of CLIP or invariant chain in class 2 MHC?
HLA-DM.
HLA-DM.
Yeah, HLA-DM. It has nothing to do with HLA-DR, DQ, DP, right? Nothing to do with that. Those are class 2 MHC. HLA-DM is part of where it comes into play in that pathway. Nothing to do with the class 2. That's about it.
Thanks,
Questions?
everyone.